on solving these 4 questions. Was it easy? Was it difficult? Could you do it, with other words? As always, nobody wants to tell me that, so let's better move on. So I put on these slides. Normally I do this at the blackboard, but this year it's not possible, of course, so we do it with slides. And as you can see in your handout, we start out with the problem that Morgan had in 1910 when he isolated the first fly which had white eyes instead of red eyes. And of course, Morgan, as you know, flies don't live forever. And Morgan knew that, of course, and he wanted to make a stock. A family of flies which all would have white eyes. So here is the question: how did Morgan and his collaborators establish a white minor stock starting from a single white-eyed fly? You know by now, or maybe you know by now, that white-eyed flies, they have a mutation in the white gene. And these are loss-of-function mutations which are recessive to wild type. The second thing which you know is that white is on the X chromosome. In both cases, Morgan didn't know. And so my question, the first question to you is, what was the sex of this white-eyed fly that Morgan found in 1910? Can anybody tell me? What do you guess? Based on the information you have above, you should be...
It was probably male because the white phenotype is recessive.
Genau. Males are XY. So if you have one mutated version, you don't have a wild type version left, which could complement. So it was most likely a male. So what would you then do with this male, white-eyed male? What would you do then?
I would collect as much female flies as possible, but then only cross this one male with all the other females to make sure that the genes you have in this male is selected.
You know, one male has one X chromosome which it can pass on. So, but you are right, you have to cross it. I mean, Morgan didn't have anything else. He had this one white-eyed fly and a ton of wild-type flies which have red eyes. So basically, he would have collected virgins and crossed it with this white-eyed male. That is sufficient. And if he then, so this would be the cross he would have established. This red eye, so this here is the candidate he found, and he crossed it with whatever virgins he could find in his other fly collection, which was wild type, basically, isn't it? So it had 2 copies of the white gene. Females are XX. And now when you make the Punnett square for this very simple cross here, you can see that the male produces 2 kinds of sperm, whereas the female produces one kind of egg, white plus. And if you then write the zygotic versions which come out of this cross, it's of 2 types, the female, which have red eyes, and the males, which also have red eyes. So all of a sudden, the nice white-eyed flies have disappeared. So question is, what do you do now? Anybody has an idea?
You could just recross the males and females from this generation, and then we will probably get with a certain probability a
let's see
female
what-
that's minus minus.
You know, so if we do that, we just take whatever flies we get out of the first cross them with each other. In the Punnett square, this is what shows up here. So we get all females are red eyes because the white plus copy is dominant over white minus. These have anyway red eyes, and among the males you have two types of males, some which have red eyes, some which have 50% have red eyes, 50% have white eyes, theoretically. What do you do then? Because this is not enough yet to make a stock. So what a homozygous stock. So what would you do now? Anybody an idea? Morgan has now the white eyes males, the white-eyed males back, but he still doesn't have any white-eyed females, which he would need to establish a pure stock. So what did he do?
He needs to cross it and not again, because now he has females which has the mutation.
But he doesn't recognize them.
But he doesn't recognize them, yeah. But in this case, it will be just lucky punch.
You
but
know,
the possibility that some of the females have it is quite high.
Exactly, you are right. So that's what I wrote here. So the males he can select, and he crosses them with whatever female he gets. And as you said, 1/4 of these X chromosomes, in principle, has the white minus allele. So after these two generations, he has homozygous females, hemizygous males, both have white eyes, and he can cross them with each other, and he has his white minus white-eyed stock. Does anybody know how would one could have abbreviated this by one generation? Do you have an idea? What did Morgan probably do? I don't know, but
maybe one would have had introduced a female that has a balancer when the balance of a balancer would die.
Yes, that balancers were not invented yet. That you don't, of course, don't know, but the balancers, the X-ray mutagenesis technique came up in the 1920s, and the balancers were not available until later, so balancers were not there yet. But to make the long story short, you could keep the male, which he originally isolated here, the red male here, and cross them to females that he got here in the first place. So he would, instead of taking, so he could have, let's see here. Yes, so here he could have collected these females and crossed them back to the father. In fly genetics, that is possible. So if you do that, then of course you gain one generation and you have your white-eyed stock already. But both possibilities are feasible. So I don't know what Morgan really did, but since males in these flies live for several months and one generation is something like 15 days, he could have reused this one male in a second cross and establish the stock one generation earlier. Everybody understood? No reaction probably means yes, that's good. So now we start with the first question you had. So I just read it to remind you what it was. A new mutation for eye pigmentation has been isolated. Its name is G. It has been mapped to the third chromosome. G has a similar phenotype as Claret, CA. Claret also maps to the third chromosome. G as well as CA are recessive and homozygous viable. How do you find out whether G is an allele of Claret? So the important information in here is both are homozygous stocks. Both have an eye color phenotype, which is very similar. And you want to test whether G is an allele of Claret or whether G is a hit in a different gene. So basically what you do is a complementation cross. Very easy. And so you are collecting males from one stock, you are collecting males from the second stock, and you are crossing them with each other here. There are two possibilities. G is an allele of Claret or G is not an allele of Claret. So they are drawn here. Does it matter whether we collect females from one stock and males from the other stock? In this case, anybody an idea? Do we have to collect virgins here from, do we have to take virgins from Claret, or could we also use males from Claret? Anybody? Nobody wants.
I think both is possible because
both is possible,
yeah,
exactly. So we don't have to. It's not on the X, exactly. That's important. So it's not the sex of the parents is not important. So what do we get here? So that, again, the Punnett square is very simple because each parent only produces one type of gamete. The same is true on the other side. So what would be the eye color of these flies here? Anybody can tell me? What is the eye color of these flies? Let me know, please. No, you know it. Come on, tell me.
Normal wild type.
Genau. These guys here. What is the eye color phenotype there?
And they would look the same as the parents.
Genau, exactly. So that means here, if the eye color of the cross is wild type, then you know G and CA affect different genes. If the eye color is identical to the parental, then there is no complementation between G and CA, and meaning G is an allele of Claret. Everybody understood? I guess so. Very simple. Complementation cross. Here, complexity increases slightly. It's about making balanced stocks for a dominant mutation Z, which is homozygous lethal. And you will see that the Punnett square is not always as simple as it was in the previous cases. So it's where one could have done it without the Punnett square also. But here you will see that things can become a little bit more involved. And then it's good to use the Punnett square to keep track of all the gametes and keep track of all the diploid progeny. Okay, so what do we know about Z? We know that an unbalanced Z or heterozygous Z stock or flies are available. That we know. We also know that the phenotype of Z is dominant, and we know that Z is homozygous lethal. Now, we assume Z is on the X chromosome, on the sex chromosome, yes. So, if Z is homozygous lethal, then this of course also means that Z, hemizygous Z males will not survive. They are lethal. So if you look at your collection of heterozygous Z flies and you don't see any males in there which show the phenotype, then this already indicates to you that Z must be on the X. Everybody agrees? I guess so. So now the question is, how do we establish this Z, this balanced stock for Z? The other assumption is Z is on one of the autosomes. And then, of course, males and females, when they are homozygous for Z, they are lethal. And this also means, of course, heterozygous Z, males and females are viable. So we have both sexes in the collection. And the question is, of course, how do we get a balanced stock then? We start with assuming that Z is on the X. And before we do that, we briefly have a look at what this balancer stock that we have, this lethal 1 over FM7, what the flies in this stock are. It's not so obvious, as I know from my experience with in previous courses. That's why we have a brief look at what the possibilities are. So these are all the possibilities genotypically spoken that are possible, and you should tell me what the sex of the different genotypes are. What would the genotype of this here be? Anybody knows?
Isn't L1 like a lethal marker, so this genotype wouldn't even be viable?
That's true, it's lethal, but would it, if it would survive, would it be male or female?
Female.
Female. What is the sex of this fly here?
Same.
Exactly. And as a consequence, this is also a female. So all these here are females. These here are the males. That's because all these here have two X chromosomes, and these here have an X and a Y chromosome. So they are the males. Since the male here is hemizygous for the lethal mutation on the X, it's of course lethal, as are the homozygous lethal guys here. So FM7 over FM7, balancer over balancer, the X chromosomal balancer is viable, but it is sterile. We will come back to that later. Why does that have? Why does this have to be the case? Is this not lethal but viable? And importantly, why is it sterile? Then we have the two remaining, which are left, so lethal 1 over FM7 females and FM7 over Y males. So when we cross these with each other, like shown on the next slide, here is the Punnett square, the female gametes, male gametes. We get lethal 1 over Y, which are males, but they die. We have lethal 1 over FM7, and we have FM7 over Y. Females, males, both viable and fertile, and we again have here the homozygous FM7. They are viable but sterile. But again, what comes out here, the guys which are viable and fertile are exactly the same as the parents, meaning we have a balanced system, we have a balanced stock. These kind of stocks in the lab, you keep them, and whenever you need flies from such a balancer stock, you go, you keep it, and you go and collect virgins or you collect males for a cross you are planning to do. Everybody understood how the X chromosomal balancer stock works. If you have questions, you should, of course, let me know so we can come back to it and discuss it. But since there are no questions, we go on. So how do we make this balanced stock for Z if Z is on the X? We said that males are not around, they are lethal. So we have to collect virgins from that cross stock, which is not really a stock, rather a collection of heterozygous flies. And we cross them with the balancer because we want to introduce the balancer. And the Punnett square is simple. Male produces 2 types of sperm, Y chromosome only, FM7. The female also 2 types, like indicated here. You make the diploid combinations and you indicate what the phenotype is. And that is possible. FM7 has the dominant mutation bar, so we can identify it. We have Z, which we can see, it's dominant, and we see it's a female. The same is true here, except that it doesn't have Z. These guys are again lethal, these are wild type. So we are not there yet. From this cross, we have to select these virgins here. So we collect virgins from this cross, this type here, and cross it again back with males, like here. These males, again, we obtain them from the stock, and we cross them to these virgins. And if you look here at the Punnett square, what comes out of it is exactly what we put in, the FM7 males and the Z over FM7 females. So we have established a balanced stock. If we realize, because we realized Z hemizygous males are lethal, so we knew it must be on the X. If Z over plus males are around, we know it's on the autosomes, so we have made that decision. We know it's on the X if there are no males, and we have established a balanced stock in 2 generations. That's how it usually is. And the other thing to remember is the X chromosomal balancer stock is a little bit more complicated in a way than the autosomal balancers, because we are talking about the sex chromosomes. Now here, I mentioned that it is important that FM7 over FM7 is sterile. Here, these flies, of course, show up. And if they would not be sterile, imagine that they would be fertile. It's very likely that we would slowly but almost surely lose Z, because these females here could mate with these males. They are the only ones in the stock. And chances are quite good that after a while, we do not have these females here in the stock anymore, and we have lost Z. That's why FM7 over FM7 has to be sterile. It cannot be lethal, because then these males would not survive. Because these males then would have, if these here are lethal, then of course FM7 over Y is also lethal, and it doesn't work. So that's why the X chromosomal balancer chromosome, when homozygous in females, is sterile. Everything clear? Okay. So now we move on to the autosomes, and I will discuss one situation where we assume we assume Z is on the second or Z is on the third, and in both cases we have crossed it with the second chromosomal balancer stock, which in this case is sternoplural over curly. So we do that. Z is on the second, so curly is the balancer for the second, so we have to look at only the second chromosome. We can't forget about the X chromosome, we can't forget about the third chromosome, we can't forget about the fourth chromosome, and the Punnett square is easy. Each parent generates 2 types of gametes, so we get 4 types of progeny, and importantly, they can all be distinguished phenotypically. These here are Z sternoplural. Sternoplural is the dominant marker here on the second. These here are Z and curly. They are only sternoplural. They are only curly. If we do that now with Z on the third and curly on the second, we of course have to follow the second and the third chromosome in the Punnett square. And that looks then like this here. So we have to look at the second as well as the third. Also for the female, the Punnett square remains fairly simple still. And by looking at the phenotypes, you can see that we have exactly the same phenotypes as we have over here, independent whether Z is on the second or Z is on the third. So meaning that independent of the position of Z, the resulting phenotypes are exactly the same. Hence, it is too early to conclude anything about the position of Z. This implies we have to do another cross to find out where Z is mapping on the second or on the third. That is on the next slide. So here, from these crosses here, we collect these flies here because we want to combine Z and curly, Z and curly. We don't know that this is already it, but we will know it after the next generation. But phenotypically, these flies here are exactly the same as these. We do not choose these because sternoplural is not the balancer chromosome. It's just the dominant marker on a wild-type chromosome. So curly is the balancer. We want to introduce, to combine Z with the balancer chromosome. Okay, so from both crosses, we have isolated Z over curly, males and virgins, and we cross them with each other. And when Z is really on the second, things are very simple. The outcome is we get only Z over curly flies because curly over curly is lethal, Z over Z is lethal, and we have a balanced stock. The only genotype, phenotype emerging are Z curly flies. If we see that, we know we have mapped Z to the second chromosome. Now, the Punnett square for the other cross is quite a bit more complicated, and You have to make sure that you don't make any mistakes. I hope I didn't make any. You get 16 different genotypic classes, which result in 4 different viable phenotypes. Some of these are lethal, as you can see, homozygous curly or homozygous Z here. They are, of course, lethal, but in this case here, you, of course, get the Z curly flies phenotypically, but you also get flies which are only Z, or only curly, or even wild type. And if you see that you get these phenotypes here, you immediately know Z is not on the second chromosome. Everything understood? No answer means yes, I guess. So if you want, I will not do it now for Z on the third and Z on the second using the other balancer chromosome for the third chromosome. It works in analogy to what I just showed you. If you want to try, you can, of course, do that. Maybe you should, just to try out whether you get the same answer as we got here. But anyway, this is how you map your mutation if you are not sure is it on the second or on the third. It could also be on the fourth, of course, but as I mentioned in the lecture, the fourth is only 1% of the genome. You rarely happen to work with genes which map to the fourth chromosome. Okay, so we move on and come to the question number 3. And based on the few people who were asking questions on Wednesday, this was the question. This was the third question. Seemed to be more complicated than the others, so we will look at it in detail. So what is it about? It's about mapping mutations relative to each other by meiotic recombination. Of course, if you don't know what meiosis is, what meiotic recombination, then you have a problem. But I'm sure you remember from your previous lectures, meiosis is the process which is occurring in the germline of animals, where the diploid cells are dividing in such a way that at the end you have the haploid gametes. And in this process, genetic material can be exchanged between the chromosomes that we obtained from father and mother. In the fly, importantly, meiosis, meiotic recombination only happens in the female, not in the male. And that is an important information if you want to answer this question here completely correctly. Okay, so the information we have is that the suppressor is homozygous viable, and the other stock which we have to talk about is this here, Aristales, black, curved, and speck. So this is a second chromosome which is marked with 4 recessive markers, Aristales, black, curved, speck, and they are distributed along the second chromosome. Aristales at one end, chromosome arm 2L, B, black is in the middle of 2L, C is in the middle of 2R, and speck is at the other end of the second chromosome. So they are more or less equally spaced along the second chromosome. And before we go into this, I have here a screenshot of this historic paper by Sturtevant. Sturtevant was one of the of the collaborators of Morgan at the time. And just to show you what the state of the research was at the time, so what I here have in red, it reads, Boveri brought forward considerable evidence from the field of experimental biology indicating that the chromosomes play an important role in development and inheritance. So, of course, for us these days, this is very clear, but at that time it was a little bit different. Things just started to happen. And Sturtevant in this paper, he could show that they had isolated 6 X-linked factors. So white was, of course, one of them, and they had isolated 5 other mutations on the X chromosome, and they could recombine them. Meiotic recombination. So at the end, Sturtevant had an X chromosome on which all 6 recessive markers were present. And he also showed that the recombination frequency between each of them was reproducible, indicating that these 6 factors were arranged in a linear manner. So whatever it was, nobody knew about DNA or its linkage. DNA was known, it had been isolated by Friedrich Miescher 40 years earlier or so, 30 years earlier, but of course nobody knew that this molecule would have anything with inheritance. But Sturtevant's data suggested that whatever it was that contained the genetic information, it was a structure which was arranged in a linear manner, where the individual genetic factors lined up in a defined order. So that was this paper here from 1913 by Sturtevant. The other thing I wanted to briefly mention, just for your information, this name suppressor is a little bit weird. You might not have heard it. So in fly genetics, you can make screens looking for mutations. If you have such a mutation, you can look whether you can find modifiers of the phenotype that you have found previously. So here we have a mutation X, which gives a certain phenotype. It's located somewhere on the 2nd chromosome. And then you ask the question, can we induce mutations in other genes which modify the phenotype of X? And thereby you can isolate 2 different types of mutations, either mutations which enhance the phenotype of X or suppress the phenotype of X. And if you find such modifier mutations, they are likely to work in the same pathway as the first mutation X that you have isolated. They are genetically interacting with each other, and genetic interaction is always a good indication that two genes have something to do with each other. So much about the explanation what the suppressor or an enhancer is. Okay, so we already said suppressor, this is a homozygous viable stock here. It doesn't have a phenotype, and I have indicating that the color is in red is the suppressor mutation. And here we cross these flies here with this other stock I have indicated in the question, Aristoles black curved speck. It's also homozygous viable. And of course, very simple, you can only get one type of progeny here, heterozygous for red chromosome, heterozygous for the black chromosome. I have written here the 4 pluses because, of course, on the red chromosome, the Aristoteles, black, curved, and speck loci are not mutated. They are wild type. So wild type Aristoteles, wild type black, wild type curved, wild type speck. And since it's heterozygous now for Aristoteles, black, curved, speck, these flies here look wild type. And we don't know where the suppressor mutation is. We want to find where it maps relative to the others. So what do we do? You know that meiotic recombination happens only in the female, so you would collect virgins from this stock here. And as indicated in the question, you collect these flies, that's from the F1, suppressor B of X Aristoteles black curved flies are selected and crossed back with Aristoteles black curved speck. So that is exactly what I do here. Importantly here, you have to use females, not males, because there is no meiotic recombination in males. So here you have to use females. And we consider only single crossover events, and we can only identify them if the recombination, the crossover happens in three locations, namely between Aristoteles and black, between black and curved, or between curved and speck. If the recombination happens over here, let's assume that the chromosome continues here, this wouldn't make a change because we could not identify the recombination event. Only when we separate Aristoteles and black and curved and speck from each other, we can identify them, and we can identify them because we are crossing them back with Aristoteles black curved speck. The markers are recessive. That's why we have to cross these females back to these. And now the meiotic recombination occurs in the germline of these females here. So if there is no crossing over, that can happen, of course, then the situation is simple. The male produces only one type of sperm, since it's homozygous. The female produces two types. We get two types of progeny, wild type and Aristoteles black curved speck. And if we test for the suppressor activity, of course, this one here, these flies here have not obtained the red chromosome, so they cannot contain the suppressor. So the answer here is no. These flies here, of course, have obtained the suppressor chromosome, and the answer is yes. So now, how does the Punnett square look like if we include the products from meiotic recombination? That is shown here on the next slide. And for every single crossover that we consider, there are two recombination products. If we make the recombination here in position A, we get these two egg plus black curved speck and Aristoteles plus. In analogy for crossover at position B, crossover at position C. So this is, in addition to the parental chromosomes, you get 6 additional egg types which represent the recombinants, the meiotic recombinants. The male, again, has only one type of chromosomes, and so if you get the diploid, you have here these guys here, which are now heterozygous for Aristoteles. And if you do that now for all the others, very simple, you get the 6 different type of flies, which are which you obtain because meiotic recombination has occurred. They are different from what you get in the previous, on the previous slide, if no recombination occurs. Now here, the phenotype, who can tell me what the phenotype of these flies here are? Is anybody?
BCSP.
Genau. Very simple. They are homozygous, so black curved speck. What is the phenotype of this fly here?
AL.
Genau, Aristoteles. Deep top. And I'm sure you have understood. These are curved speck. They are Aristoteles black. They are only speck. They are Aristoteles black curved. So that's very easy. Now the next column here asks what is the activity of suppressor, and who knows what the activity of this chromosome is here? Anybody can tell me? Of course, you cannot know, but you can see it on your question. In the question, there is a table. And in that table, it says that flies of phenotype black curved speck, they have suppressor activity. So did you see that in that table? And if this one has activity, of course, it also means that this recombinant here cannot have activity. And you can get all the information from the table. So I didn't, you couldn't know that, but it's indicated in the table. So that's the yeses and nos in that table have been filled in here. And now you can, basically from these answers here, you can tell in which interval of the red chromosome the suppressor is mapping. Anybody can tell me where the suppressor is mapping?
It should be after C, because as soon as the last part of SP is lost, the suppressor activity is also lost.
Yes, but can you can be even a little bit more precise? If you, I mean, you were talking about these guys here, isn't it? After C, you said. Now move one up. I mean, the recombinant which has the least red chromosome and has activity is the one up here. You agree?
Mm-hmm.
Okay, so with other words,
I mean, it has this part that includes the SP allele, like this the not recombinant SP allele.
yes,
so if we move further
sure.
down, we slowly lose like the next chunk. So first we lose like AL, then B, then C.
Absolutely, but the smallest interval, you see, of course, the whole red chromosome has it. And then this here, of course, also has it. But then you can have it even smaller. This is also yes. You see, the answer is yes for this one. But for this, it's still yes. You see?
Okay, I just thought other way around.
Okay.
Like was looking at the left.
Okay.
The wild-type chromosome.
Okay. Okay. You were going for the no. But of course, now I understand. But of course, we want to know where this one doesn't have the activities, you see? But this one has it. So this here has obtained the smallest part of red chromosome, which still gives you suppressor activity. Understood? Is it clear?
Yep.
Okay. So how do we get these answers here? That is shown on the next. So in the question, it's written that you have to test the recombinants to see whether they have this suppressor activity. I wrote these were subsequently tested for their suppressor activity by crossing them with X over balancer flies. So that is what we do here. So we have the recombinant, which are heterozygous for the Aristolase black curved speck chromosome. And we ask, has the recombinant chromosome suppressor activity? And we cross it with, and we take males here. Why do we take males? Anybody can tell me? Males do not have meiotic recombination. If we would take females here, we would be in danger to lose the suppressor again if it has it, you see? So that's why you have to use males here, because they don't show meiotic recombination, and so you do not change the integrity of this chromosome here. So you cross them to these females. Again, the Punnett square is fairly simple. The curly flies are not of interest to us because they don't have X. This chromosome has the X mutation, which gives you the dominant phenotype. And then you get two types of progeny, X over recombinant and X over Aristolase black curved speck. Oops. And the question is then, do we see the suppressor phenotype in the progeny or not? If we have only flies with phenotype X, then of course there is no suppressor activity on the recombinant. If the ratio of flies which show the X phenotype and the suppressor of X phenotype is about 1:1, then we know that the recombinant has the suppressor activity. And this gives us the information to fill in here, yes or no, or yes or no, and yes or no, because we chose males here, these males, these males, these males, these males, these males, these males, and cross them in the way I showed you on the next slide to find out whether it has the activity or not. And that is how, that is basically the proper solution of this problem. One last thing I wanted to mention, why do we have to take single males here? One could also say we take just all the males which have this phenotype here and cross it with these virgins. Why is that not a good idea? Anybody can tell me? This is a slide I just introduced before this lecture here, so you don't have it in your handout. But let's look at the situation, and then it will become immediately clear to you. So Aristolase and black are fairly far apart. So this suppressor mutation could either be here or here or here. And then it depends, of course, where the crossing over occurs. The closer suppressor is to Aristolase, the higher the chance that our black curved speck chromosome has the suppressor activity. That is as we had it in our question. There in the table it says black curved speck gives you suppressor activity. The answer was yes. But now, since the distance between Aristolase and black is fairly big, the suppressor could also be closer to black. And then, all of a sudden, the black curved speck chromosome, which according to our table should be yes, doesn't have the suppressor activity anymore. You see? So that's why here you have to use single males to do these test crosses, because among the Aristoteles black speck, if you test 20 different black curved speck males, it could be that some of them are suppressor or not suppressor, depending on where the suppressor mutation is located. Since all in our question, all black curved speck males seem to have suppressor activity, suppressor must be very close to Aristoteles. Is that clear? Why we have to take single males for that test? If we do not do that, we could get one male, we could have different type of males which have different activities. That would be a problem. Okay, let's go to the fourth question. That has again something to do with meiosis, and the key word is X chromosome non-disjunction. And this observation of X chromosome non-disjunction was the topic of a paper by Bridges, which he published in 1916, and it was kind of a proof of the chromosome theory of heredity. So I briefly mentioned to you that it was not clear at all what the chromosomes are doing, and then at one point people realized that Mendel's formalism and the behavior of chromosomes during mitosis and mitosis was kind of suggesting that the chromosomes could be the bearers of the hereditary material. And this non-disjunction experiment was part here of this paper by Bridges, and the title is Non-Disjunction as a Proof of the Chromosome Theory of Heredity. So on the next slide, we go through just a part of the Bridges observation, based on which he concluded that the chromosomes in fact were the bearers of the hereditary material. So the question says that we have red-eyed males, and since you know they are white plus means we have to consider the sex chromosomes. The males are X over Y. And these males here are crossed to white minus females. And the females produce one type of eggs normally, and the males produce two types of sperm. And if you combine them, you get red-eyed females. White plus is dominant over white minus, and white-eyed males. Now, if the X chromosomes in the female are not properly distributed to the daughter cells during meiosis, it can happen that both X chromosomes end up in one cell and none of them ends up in the other. So this is indicated by these two lines here. Both X chromosomes are in one egg. No X chromosome indicated by a 0, null, in the other egg. And now if we make the combinations, you can see that the diploid combination, we have now a female which has 3 X chromosomes, and we get males which have only 1 X chromosome, no Y chromosome. And we have here another class which has a Y chromosome and 2 X chromosomes, and we have a class which has no X chromosome, but only a Y chromosome. So what are the phenotypes? That's indicated here. Triple X here, that would be female, and I write here but lethal, but the information, I never did this, of course. This is from textbook. So I have conflicting information. People, some say it's lethal, some say it's not lethal. So I don't know what it is. But if they would hatch, they would have red eyes because they have a copy here of white plus. Then we have the females which we do not expect because they have now red eyes. These here would be the same as these. These here are female. They have white eyes. They are fertile, but they are rare. Only about 1 in 5,000 you find them. That's, again, according to the textbook. Then you have null. Yes, null over X, genau. These are these, huh? Now these are males. They have red eyes. They are the other rare class that we find, 1 in 2,000. They are sterile. So these we find, as indicated in the question, very rarely. We do not find these here because they do not have an X chromosome. So no X chromosome means dominant lethality. So you will never get these flies here. You probably wonder why I tell you that these here are females and these here are males. I mean, you probably remember from your previous genetic classes that in humans, if you have a Y chromosome, then you have a good chance to end up as male. And if you don't have a Y chromosome, you have a good chance to end up as female. So this indicates to you that something happens a little bit different than in us. And that is here summarized on this slide here, the role of the Y chromosome in sex determination. So human, mouse, Drosophila, XX gives you female, XY gives you male, X null in flies, male, sterile, XXY, female, fertile. Situations are different here in mouse and human. There, whenever basically you have the Y, because the Y chromosomes contains this SRY gene, which makes you male. In the fly, that doesn't seem to be like that. The Y chromosome doesn't contain an SRY gene, and it contains a number of genes which are required for fertility. So that's why these males here lacking a Y chromosome are sterile. So the question is now, of course, what is the difference between these here and the flies? Why is the Y chromosome not important? That is explained here. I don't know how much you know about sex determination. I don't know much either, but it can be very different from animal to animal which you are looking at. And I just mentioned in us, it's the Y chromosome which determines basically whether you are, if you have it, you are male. If you don't have it, you're female. In the fly, it's different. The fly measures the ratio between sex chromosomes and autosomes. So meaning the fly, the female has 2 X's and has 2 sets of autosomes. So the ratio is 1. 2 to 2 gives you 1. That means you become female. In the male, you have 1 X and 2 sets of autosomes. So the ratio is 1 to 2, 0.5. If the ratio is 0.5, you will end up as male. And where is it read out in the genome? There is a gene called sex lethal. And the regulatory region of this sex lethal here can sense what the ratio is, 0.5 or 1. If it is 1, sex lethal is turned on and you become female. If the ratio is 0.5, sex lethal is not active and you become male. So you can see that this is quite different from what you probably knew from sex determination in humans. And people have looked at that in great detail, and that is summarized here in this table. And you can see that the ratio of X to A can be varied in the fly and gives you different types of phenotypes. So if the ratio is below 0.5, you become male. If the ratio is above 1, you become female. If it's in between, you have a fly which is phenotypically male or female. So much about sex determination in fly. And do you have questions? Doesn't seem to be the case. So I promised you in the lecture that we will briefly look at the crossing scheme used by Jani Nüsslein and Eric Wieschaus when they were doing their very influential screen looking for embryonic lethal mutations, looking for genes, with other words, which have an important role during embryonic development. And they obtained, for their work, they obtained the Nobel Prize 25 years ago. So this was the slide that I showed you, and I will conclude this presentation by quickly going through the crossing scheme and give you the explanations why they did it like this. What I should say is, to start with, they had to do the screen for every chromosome independently. So they did a screen for second chromosome genes which have a role in embryonic development. They did a screen for third chromosomal genes, and they did a screen for X chromosomal genes. We look here at only one of the three for a screen for third chromosome mutations. And so this is the first cross. So they were feeding EMS here. This is a chemical mutagen. They were feeding these males here with EMS at the concentration where, in average, 1 mutation is induced per chromosome. Of course, this is important. If you have a too high concentration of EMS, you induce too many mutations, and then this, of course, can complicate the analysis of your screen. So let's assume we have 1 mutation here on this particular third chromosome. It's indicated in green. The other one is still wild type. These males here, after they have been fed with EMS, they are crossed with these males here. DTS, I have seen that one of you calls himself DTS. That's not him or her. It stands for dominant temperature sensitive lethal. And so if you have this chromosome, if you have obtained it from your parents and the cross is kept at 29 degrees, you will die. So you will see that using these chromosomes makes analysis of the crosses and collecting flies a little bit easier. That's why they used it. So the Punnett square is simple. The female produces 2 types of eggs, the male 2 types of sperm. And the diploids look like this here. And since the cross was kept at 29 degrees, you already reuse the progeny by 50%. You do not get these genotypes here in the progeny, but you get these 2. You cannot distinguish them because this here is a recessive mutation, and you will not see it, so you cannot distinguish these 2. But what they did is they selected males here, most likely, maybe also females, but most likely males. Some of them carry the mutations, others do not. The important thing is you take these males now and cross them individually. Can anybody tell me why you have to cross them individually and not 2 or 3 or 4 males together in the same cross? Anybody an idea?
Because the mutations could be on different spots in the chromosome. If we just cross them all together, we just lose the overview.
Exactly. We have to use only one, because then we look at exactly this chromosome and not another. The other chromosome we obtain is the TM3, but that has a dominant marker we can select against. So that is shown here in the next slide. So we cross these single males again back to the same stock as we had before. So we have been collecting virgins from this particular balanced stock here. Again, the Punnett square is simple. Each produces 2 types of gametes, and these here are lethal because they have the dominant temperature sensitive. Homozygous balancer, as you know, is also lethal. So the only viable flies emerging here are these. So we already have a balanced stock. That is why they were using this dominant temperature sensitive lethal. It makes fly crossing much easier. Here you do not have to select for these here, because what do you do with these? You cross them on. Now you have your stock in which the green chromosome has been more or less, you would say, cloned. It's the only non-balancer third chromosome which is present in the stock. Now, they were, of course, selecting. They have established, I think you remember from the previous slide, they selected 27,000 families, so quite a good number, and looked for those where M over M was lethal here. You see? So if you cross these with each other, you get, of course, the parental guys again, like here. These die. And the question is M over M viable? If it is, you can discard it because it's not interesting. They were looking for embryonic lethality. So if it's viable, if you get adults, it's not what you wanted. And you only keep those where M over M was lethal. That is easy to see. If you have flies which have the balancer here, the dominant marker, then you know they are there. If there are non-balancer flies, you also know it's viable because then they are M over M. So they kept all the guys which were lethal, where they had only heterozygous progeny here, and tested, do we see now changes in the patterning of the embryo? And that is how they selected all the different zygotic alleles, the pair rule, the gap genes, the segment polarity, and so on and so forth. That is, in a nutshell, the screen, the famous screen of Nüsslein, Volhard and Wieschaus, and I hope you can appreciate it now how they did it. Anybody a question? Doesn't seem to be the case. I understand that because, as usual, I took a while, but normally, actually, if I do it on the blackboard, it takes 90 minutes, so we did it in 80 minutes. That's quite good. So this is the end of the Affolter course. I hope you had a little bit of fun, maybe not. But anyway, I wish you a good continuation of this block course and all the best for your future. See you. See you.
Bye.